right, good morning, everyone. So I had a couple of people notice that listening to the last recording, the audio was a little bit garbage. I'm looking into ways for possibly cleaning that up. It looks like my laptop defaulted to using the mic that's in the webcam that's built in instead of the actual setup. So the audio wasn't great. I'm going to see how much I can get back, but if you have questions when you're listening through it, just let me know. And I have now fixed it, so it should be much better from here on out. Sometimes it just does that. I have no idea why. But today we're going to actually wrap up the last part of Chapter 4 here, and we're going to talk about some more of the methods that we can use to investigate the stuff that we're interested in this course. And we get to start with one of my favorite topics, where we look at how we investigate heredity, which is how we figure out how much of a particular trait or behavior of interest is influenced by our genes. And there are a couple of different ways that we can do this. One of the simplest ways is a family study. And so this is going to look at how, basically you're going to start with like a family tree. If you've ever done an intro bio course and they've made you do pedigrees, it's the same basic idea, but with a human family tree. And you would look at your pedigree and you would see, okay, how many individuals in this family all have the trait of interest. So if we were looking at something like anxiety, we could see how many of those circles and squares on our pedigree are filled in, showing that they have that trait. And then we could look at, okay, are people who are more closely related more likely to share a trait than sort of random other people? The thing we have to worry about with these family studies and with a lot of other studies that we can do is that the effect of our genes is often confounded by our environment. So people who live together might share genes, but they also live together. So they have a very similar environment. So in this case, we can't get a clean, separate picture of what comes from genes and what comes from environment because we can't separate those out. And most of these, actually pretty much all of these studies, especially in humans, are going to be correlational approaches because you can't decide who gets assigned to which family and who shares genetics. You just have to measure it. So we have a bunch of limitations here, but when we first start with genetic studies, and especially before we had a lot of the techniques that we do now, they had to rely on these kinds of studies. One of the biggest ones, and I don't think the textbook talks about this, but when they were trying to look at the heredity of schizophrenia, they were looking at populations in, I believe it was Iceland, just because they have a fairly closed population, minimal immigration and emigration, and so they were able to look at people who lived in the area. They had well-known sort of relationships, they had a good map of the population, and they had really nice records with the hospitals as to who had been admitted to hospitals or treated for symptoms relating to schizophrenia. So that's one of the first ways we conducted family studies. And the methods nowadays aren't too different, but we can kind of add on them and make them better by doing other things as well. And at the bottom there, we just sort of make a note that if we've measured our variables using a numerical scale, we could report our results as a correlation coefficient. So we could report a correlation between if someone, or how likely someone is to have a disorder that they're related to someone else who has the same disorder. We don't get into the calculations of that, and that's fine. But I will explain what correlation coefficients look like in case you see them. I have taught correlation coefficients in a whole bunch of classes this year, so I don't think we've covered this in this class yet, but if we have, just let me know. But the way that I explain correlation coefficients is imagine a number line, and this number line ranges from +1 to -1. And right in the middle is 0. And if we have a 0 for our correlation, then it means that there's no relationship between the two variables that we measured. If we're at either of the extremes, if we're close to +1 or if we're close to -1, that means that the two variables that we measured are very, very strongly related to one another. So the further away from 0 we are, the stronger the relationship. And we can have positive correlations and negative correlations, whether we're closer to +0 or +1 or -1, but the positive and negative doesn't tell us anything about strength. It tells us about the direction, the type of relationship. So a +1 correlation means that we have a very, very strong, the strongest correlation possible, and the relationship is that as one variable increases, the other variable also increases. So if we were looking at a correlation between height and weight, taller people also tend to be heavier because there's more mass to them. They are just bigger people overall. So we would see a positive correlation between those. So it's not a perfect correlation. It's not a absolute where all taller people are automatically heavier than someone shorter than them. But we would say maybe there's a positive 0.7 correlation between those, which means that we'd have, so for you guys going this way, a positive, so as we increase on the x-axis, we increase on the y-axis. With taller height, we have larger weight, and the dots on a scatter plot like that would be pretty close to a line because they have a strong relationship. If we were looking at things that are negatively correlated, it's the same concept, but the relationship is opposite. So as one variable increases, the other one decreases. See if I can think of a good example for that. Did I give myself one? I did not. Okay, so we can go with the more time, or the more you miss class. So the more absences, the lower your grade would be. That's the standard one that you'll see in textbooks. So as we increase on one axis, the other axis will decrease, and we end up with a line on the opposite angle. So that's a negative correlation. And so if we see a number that's close to 1, whether it's positive or negative, we can say, ooh, there's a really strong relationship there. If it's really close to 0, it's a very weak relationship. There might not be anything at all. So we like the larger numbers, but we don't always see large numbers, especially as we start looking at things a little bit more abstract, like psychological traits. So when we start getting into things like personality traits, we might see a correlation score of 0.1 or 0.2, which is a fairly weak correlation, but those are usually statistically significant because we can get the same numbers in lots of different samples, in fairly large samples. So even if it is a weak correlation, there's lots of other noise involved, we still care about small correlations. It's all about interpreting what it means for us. So I guess the best we'll ever see in actual data, the best at least I've seen in actual data when looking at correlations in studies, is about a 0.7, 0.8. We tend not to get anything higher than that, especially for behaviors. But that's one of the major ways you're going to see these relationships reported, is what's the correlation between, or how does the rate of incidence of a particular trait differ based on degree of relatedness? Is it positive or negative or whatever? So that's family studies and a very quick crash course on correlations. Next, we can look at twin studies. And so we talked about the fact that with family studies, we have that extra confound where, yes, they're genetically related, but they're also probably sharing an environment because they're living together. And if you remember back to probably first year bio or even first year psychology, when we talk about getting our phenotype, the expressed trait of interest, whether that's expressing a disorder or having blue eyes or whatever trait we're looking at, our phenotype, that expressed trait, comes from a combination of our genes and our environment, the stuff that's acting on that. And then there's also a little bit of error left over that we can't necessarily attribute to either, but we tend to ignore that. So in our family studies, we had an issue where we could look at the phenotype, but we couldn't decide how much credit we gave to the environment versus our genes. And our twin study helps us start picking apart some of that by making some assumptions about what we're looking at. So with twins, we know that there are two different kinds of twins. You have identical twins and fraternal twins. Identical twins, they come from a single fertilized egg that happens to split after it's been fertilized and turns into two separate people with identical genetic material. That's really convenient because now they don't differ genetically at all. So any difference between our two twins would come from environmental differences. And because we have that second kind of twin, because we have fraternal twins or dizygotic twins, they're born from two separate eggs that are fertilized at the same time. So they're not identical, they're the equivalent to regular siblings who just happen to be born at the same time. So they only share 50% of their genetic material. And so we have an interesting case where if we can look at twins, we make the assumption that they share most of their environment. They tend to be, well, they're automatically going to be the same age. They usually share the same room. They usually go to the same school. And at least when they're younger, they tend to have a lot of the same hobbies and activities and stuff like that. So we get to assume equal environments, which is really convenient. And then any of the differences between our fraternal twins and our identical twins, that would be based on whether they're 50% genetically the same or 100% genetically the same. So we can do some fancy math in the background and figure out how much more likely are we to see things shared between identical twins than fraternal twins. And here, instead of using a correlation, we use something sort of similar. It's called a concordance rate. And instead of reporting sort of how, how do those two measures relate to one another as they change over time, the concordance rate is just looking at how likely are two people to have the same trait. So a concordance rate between fraternal twins should probably be lower than those of identical twins if the trait of interest is genetically related. So that means that identical twins should be more similar for this trait than fraternal twins because they are more closely related. And if we see an equal concordance rate between identical and fraternal twins, if the difference in their genetic similarity doesn't matter, if it doesn't affect those rates, then we can conclude that it's something that's probably driven by environment, not genetics, because there's no difference between those people that differ quite massively on their genetic sameness. So that's the logic behind twin studies. And apparently I don't have a slide for it, but we can talk about the one in the middle here. So we talked starting with the family studies, ending with the twin studies, and in between we have adoption studies. And these are sort of a special, it's almost a case study, but not quite, because they tend to be on a much larger scale. But this is one of those times when psychologists make use of resources that are available to them. So you might have by now in your careers heard about the Minnesota Twin Study. And that's kind of a misnomer. It isn't a single study. It's actually a collection of data because Minnesota happened to keep really good records on all of the children who were adopted in that state. And that those records were then pretty easy to link to things like hospital records and all of the other information that might be useful for those of us who want to look into biological traits. And so researchers were able to go in and get access to those files and contact some of these people who had been adopted and chat with them about sort of where they had ended up and get them to fill out surveys and stuff if they agreed. And the Minnesota Twin Study is a fancy one because it wasn't just a twin study and it wasn't just an adoption study. It was a neat hybrid of both. So let's step back and talk about what an adoption study lets us look at, and then we'll talk about how we can hybridize the two. But we're going to go back to that idea of our phenotype, our expressed trait is a combination of our genes and our environment. So if we look at adoption, we have children who are sort of separated from their biological parents, the people that they share genes with, and they are moved to a new family where they share an environment, but not genes. So this lets us look at, okay, how similar are these children who have been adopted to their adoptive parents? And how similar are they to their biological parents? And in this case, they don't share any environment with their biological parents. They only share genes. And they don't share any genes with their adoptive parents. They only share environment. So we get to pull apart if they are more similar to their adoptive parents, then we assume a stronger environmental effect. And if they're more similar to their biological parents, we assume that genetic effect. And that's where our twin studies come in, because we add that extra layer to it. Well, what if we have identical and fraternal twins who are adopted? And what if they're conveniently adopted into different homes? So we can look at twins who are in the same home versus twins that have been separated, so that we can have how much of their environment do they share. If they're in the same home, you kind of assume the same environment. If they're in different homes, they have completely different environments, no sharing there. And if they're identical twins, we know that they're 100% genetically the same. And if they're fraternal twins, they're only 50% the same. And so you could see a nice sort of a 2x2 diagram where we could work out how much similarity do we see when they are 100% genetically the same, 50% genetically the same, when they're sort of sharing an environment versus not sharing an environment. And if you ever take a behavior and genetics course, which I have taught in the past, you get to do all sorts of fun background calculations to work out the relative contributions of genes and environment, but we can actually report that and sort of get a good idea of how much of a particular trait is genetic driven versus environmentally driven. So it's kind of a weird concept to wrap your head around, but I guess when we kind of walk through them step by step, the logic makes sense. And so again, these have to be those correlation type studies because we can't manipulate this. You can't assign people to be adopted. So all of these, when in humans, are sort of borrowed data from other resources, like that Minnesota twin database. All right, so what if we don't want to just do correlations? What if we do want to do an experimental approach? This is typically going to be done in non-human animals. Most of this would be done in something like lab rats or lab mice. And the reason that they do that is because with those models, those species that we can use to kind of give us a better idea of how stuff works together, we have really fine control on what each individual has in their genotype. So we can get genetically identical rats, and we could change, say, one gene between all of the rats in a litter. So they are identical, we know exactly what genes they have everywhere else, but then we can use genetic engineering to change one gene, or maybe a string of genes, and see how their behavior or their physical traits change with just that one switch. So there's a bunch of different ways that we can do that. And it doesn't always require the identical genes in the background. That's just one of the cleanest ways to look at it. For other things we can use a knockout technique. And so this is when we have a non-functioning mutation introduced into a gene. The simplest way to do a knockout technique is to have enzymes of some kind come in and slice out the center portion of a gene. So if you already know the sequence of a gene, you can design molecules that'll go in and latch on and cut out what's in the middle and stick it back together. And then if you're missing all that stuff in the middle, you can't produce a functional protein from that gene anymore because we're missing half the instructions. And sometimes these knockouts, they don't have to be large. You can remove a single nucleotide and cause catastrophic failure down the line, but it's usually a lot more reliable to take out a big chunk, because then you can usually actually see the difference if you run some tests in the background. But a knockout technique is fully turning off that gene, deactivating it by turning it into something that no longer produces the proteins that would make the thing happen. We can also use a knockdown technique, which is where you can silence the functioning of a gene by interfering with its expression. So we can use something like antisense mRNA, where we talked earlier about how we have our complementary strands of DNA, and when we want to make a copy of a gene, when we want to send it outside of the nucleus to make proteins from it, those strands have to separate, we have to bind to it, and then sort of make that copy as we move along the template strand. But if we can get something else that is quicker to bind and maybe block off that gene, we have mRNA that can get in there and interfere with the process of transcribing mRNA, we can keep it from expressing that gene. So the gene is it's technically functional, it could work, it's still there, but we've introduced other things that block it from working. So it's not physically damaged, it's just interfered with. Oops. Oh, and I actually included an image of that. This is, so here they're actually showing that instead of blocking to the DNA itself, they have that, okay, you can transcribe your messenger RNA, but as soon as it leaves the nucleus, we're going to have some other mRNA that's the exact opposite, stick to it and block us from turning it into a protein. So there's all sorts of steps you can interfere with that production at. So if we know the stages of going from our double-stranded DNA out to proteins, any of those stages could be messed with if we know what products are where and when. Some other interesting experimental approaches that we can use are gene transfer. So this is when you can take a gene from one organism and insert it into another. A lot of this is usually done with things like, lately we've seen a lot of, say, bioluminescence genes being inserted into other creatures. So they've found things from, say, algae that tend to glow, they tend to produce bioluminescence. And they've identified the particular genes that produce the proteins that glow. One of them is luciferin, I think, is one of the big ones lately. And so once they've figured out what it is and where it is, they can design, again, molecules that will go in, sit on either side of that gene, and chop it out. And we can use sort of background techniques to make lots of extra copies and insert it into the DNA of another species. This is a whole lot easier to do in something like bacteria. If you've ever heard of cloning, not making an extra sheep that has that is genetically identical to another, this is the more, I guess, boring kind of cloning, where you use bacteria to take up extra plasmids, which are the bacteria equivalent of chromosomes, and make lots and lots of copies for you. That's actually how we get things like insulin, or at least that's how we used to get things like insulin. I think it's a lot more not in bacteria now. I think they can do it artificially, is the term I was looking for. But basically, you can sort of steal these bacteria, you can co-opt all of their mechanisms, their ability to copy things, and then their ability to make proteins by using instructions, and you can insert plasmids that have whatever genes that you want on it. And you can give them multiple plasmids, the equivalent of giving them lots and lots of different chromosomes that each have sets of instructions. And that's how you could get a random E. coli bacterium producing insulin because you gave it the genes to produce insulin. So you can use gene transfer to do stuff like that. That's also how they do things like getting those bioluminescent genes into cats or mice. But it's a lot more complicated with larger organisms, which is why we typically stick to bacteria. But when you're doing it with larger organisms, you would have to start at the stage of sort of fertilization, a single cell, because each cell has a copy of all of the DNA that was present at fertilization. So if only one of the cells has that introduced DNA and the other cells don't, you're not going to see the thing you've inserted expressed everywhere. So if you start when there is only one cell and you modify that one cell, then it would be passed on to all of the cells that are produced by that one dividing. And then the next one is gene therapy, which is sort of targeting and manipulating genes specifically to treat disorders. So it can involve gene transfer, it can involve sort of other things like that. And a lot of this will be done usually through vectors. So they will, again, borrow the mechanisms in place in something else to carry out a function that we want. So here, they'll usually take a disabled virus. So they'll take, viruses are really good at copying themselves, and they carry around a string of DNA that they can then use to insert their own material into another cell and get that other cell to make more copies of the virus. If we take out the virus's DNA and instead insert our own genes of interest, we can use the same mechanisms for them to insert what they think is the DNA to make more viruses, but is actually the DNA to fix a protein deficiency or something like that. So we can have those insert things into our DNA that way. And again, a lot of this stuff, when we start getting into doing this on larger and larger creatures, gets a lot more controversial. And that transitions us really nicely into the next concept, which is how we're going to wrap up this topic, which is research ethics. So we'll talk about why a lot of those things like gene therapy and a lot of other things in psychology aren't typically done in humans and are only rarely done in other species as well. But if we're going to do research ethics, we're going to kind of touch on a little bit of everything all at once. I know that every psych class does a brief interlude into ethics, so a lot of this will be fairly familiar to you. And again, we're going to be getting the Coles Notes version, because you could do an entire course or 5 on ethics if you were going into psychology. So starting off with plagiarism and fabrication. As students, you've probably had the idea of plagiarism drilled into your minds from a very young age, where you're not allowed to copy. You can't misrepresent someone else's work as your own. So plagiarism is just the theft of another's work or ideas. And that's why in sciences, we have a really strong pressure to cite where information has come from. So when you're doing an assignment, if you talk about, oh, somebody has done research that showed that this is the case, you can't just say that, oh, this is the case. You have to tell us why you know that. Where did it come from? So you would cite the paper that told you that. And that's why you want to be able to point to where stuff came from so that we can then think critically about things that we're reading. So that's kind of another of those things we're going to be considering in our assignment, where when you're looking at sort of this media article, step 1 is, did they actually cite the paper they were talking about, or did you have to go hunting? If they're good and they've done their due diligence, they should give you a direct link to what they're talking about. Whether they've done a good job paraphrasing it is another question. The closely related idea to this is fabrication. So instead of stealing someone's ideas, this is fabricating or making up ideas. And our textbook argues that this is possibly even more serious than plagiarism, and it's because it's introducing false knowledge into our knowledge pool. So with that concern over citing information and telling people where we got things, when you start getting into research in a particular field, you find that it's almost like a web of interrelated studies. So I've been working on publishing one of the papers from my PhD research, and I have citations to older work in my lab and other related labs, and they've cited some of our work in the meantime. And so there's this interconnected web of everyone sharing information and building off of what's been done previously. Every research paper that I've ever done starts with a literature review telling the reader what's been done in the field before, where did this information come from, and what are we going to do to add to it? But what if one of those building blocks that you're using wasn't actually true? What if it was fabricated? So if somebody made up data or intentionally misrepresented facts in order to get something published, well, then that kind of throws fault into all work that comes after it. This is why it can be more serious than plagiarism, because it can spread and cause problems in a lot of different places. So it introduces that erroneous information to the field, and one study showed that up to 2% of researchers admit to falsifying some part of their study at some point in their careers. And 2% is a fairly low number, but if you think of how many papers are published every single year, it's kind of scary to wonder, how much do we trust the research that's out there? And so hopefully the process of going through reviews and getting approved to publish in journals and having reputable journals is a good thing and helps us have a little bit more confidence, but there's always the chance that that's snuck in. All right, so the next thing we want to talk about is ethics, more on the side of what we think of for ethics, which is how do we decide what we can and cannot do in research. And as per usual, the text divides it up into humans and non-human animals. So when we're working with humans, it's a little bit different, because with humans, you can tell them what's going on. And so they focus in on one of the biggest things being informed consent. So individuals need to be told, what are the risks of participating in this study? What are the possible benefits of performing in this study? And sort of what do I need to know to make an informed decision? So an individual has to give voluntary agreement. They have to consent to it, but they have to have all the information required to make it an informed decision. That's why it's called informed consent. We can run into issues where some groups can't give informed consent. If you do research with children, children might not have the capacity to understand what's going on. And because they're not legally adults, they don't have the authority to make that decision. So instead, as a researcher, you would have to get consent from a parent or guardian. And then there's an extra step there, because with children, you're also, if possible, going to get their agreement to participate in the study. So the parents need to say that it's okay, and they need to be informed of all those risks and benefits, but the children themselves need to say it's okay too. And we have issues with informed consent where we can have bits of, say, feeling pressure to take part, or the next thing is, what if deception is involved? Because you might be thinking, if you have to tell the individuals everything about your study, if you have to tell them the risks and benefits, you might have to give them information that would then bias their behavior in the study. Humans are really awful research subjects, because if they think they know what's going on, their behavior can change to either try and match what they think you're trying to study. So they might behave in a way that they think will sort of give you better results, or they might be jerks and they might behave in the exact opposite way. So how do you prevent that? Well, maybe you don't give them enough information for them to figure out what the study's about. And that kind of bothers that idea of informed consent. So where do we fall on this? How does this work ethically? So sometimes deception, or using misinformation or not disclosing some aspects of a study, can be deemed okay. And this has to go through an ethics approval board, where they have to decide if it's justifiable to withhold some information. When I've worked with people doing human studies, there's almost always some level of deception involved. So for us, we'll have participants come in and listen to birdsong. And so we will tell them, for the informed consent, that they're going to be listening to sounds, and they're going to be categorizing the sounds into two groups, and they're going to be doing this on a computer using headphones. The risks associated are those associated with sitting in front of a computer for a long period of time, those for using a mouse repeatedly, and those for having headphones on. So none of that interferes with our study. But they don't know specifically what they're listening to or what they're categorizing it based on. We've withheld some of that information, but they know everything relevant to the risks of what they could be exposed to. We then have to come through at the end, give a follow-up where we explain that, okay, you've done the study now, here's what was really going on. And that process is called a debriefing. So part of our ethics approval is contingent upon giving them a debriefing and telling them exactly what information was withheld at the beginning and why. And for something like, we were getting you to listen to bird songs and we wanted you to classify them based on the location that they're from, telling them that information might have biased their choices, but not knowing didn't introduce any extra risks, so it was deemed an acceptable deception. And so there's lots and lots of guidelines on what you can and cannot withhold. Most of the studies that I've seen done tend to have sort of a blanket approval as long as you don't deviate from we're getting people to listen to 2 sounds and grouping them into 2 groups. But if you had to do something where there's a risk of physical pain or emotional distress, that's when it gets a lot more serious. And each study would have to be approved and evaluated based on the level of risk there. So this third party, this internal review board, would have to stand back and evaluate this from an unbiased standpoint and decide if the potential risks outweighed the sort of benefits that could come from the study. Okay, so that's with humans, but what about non-human animals? And so I've already kind of mentioned the fact that using non-human animals in research can be really useful. So I already dropped the term of model species, where you can use things like lab rats, where we know absolutely everything about, say, their genetic background. If we go with a particular strain of lab rat, we know their exact genotype at every point in their genome. So it can be really beneficial to be doing something like that. You could also be working with something much smaller. Maybe you're doing genetic work and you're using nematodes because they're only a couple of centimeters long and easy to house, and they have just enough genes for your interest. So the idea there is that you can use these non-human species to get some idea of how this might apply to humans if that's your end goal there. So some people might feel that semi-risky experiments could be more ethical when performed on animals. They'd prefer that you test a new anxiety drug on animals before you test it on humans, or something like that. Though there's been a lot of push lately looking at is it actually necessary to be doing that, and how much animal testing should or should not be done. That is a much larger issue than we can cover in this course. But we can sort of hit on a couple of the high points. One of the terms you might see thrown about is speciesism, and that's a term coined by animal rights activists talking about how humans are animals too, and by them focusing on non-human animals versus humans, there's sort of that speciesism, that focus on us being better than them or different from them. And you might even notice that our textbook talks about human research and animal research. The non-human animal thing is an addition by myself here, based on what terms are more commonly used nowadays. So it's a very constantly evolving and changing scope here. But one thing that we can know is that a lot of our animal research guidelines tighten over time. And I've even found this in my own career. As we move forward and as we sort of gain more and more technological advances, it becomes viable to do things like run a computer simulation instead of running an actual experiment. So in one of my research studies, we ran an experiment using the black-capped chickadees. We exposed them to 2 different sounds and asked them to put them into 2 different groups. We had someone do the same thing in humans to listen to 2 sounds and put them in 2 groups. And then we had a computer program listen to 2 different sounds and put them in 2 groups. And then we could compare performance between the humans and the chickadees and the computer. And wouldn't you know it, the computer did almost exactly the same as how the chickadees did. And if we can show that we can build a computer model that uses the same features as our chickadees did, then maybe we can run future research using more of those models and less birds. So we can bias our methods towards something that's less invasive, less sort of inconvenient, and do more sort of non-ethical or not ethically gray stuff. So there's lots of change here and lots of debate here, and it's continually changing as time goes on. Even within universities, if you have ethical approval for a particular type of project, you still have to renew it every single year, even if your methods don't change. And so I found that every single year we've been doing the same kinds of studies, but we have to make adjustments to our methods to show that we're taking those new regulations, those new rules into consideration as we move forward. So it's not as willy-nilly as people might think when they think about non-human animal research. Okay, so that's our very, very brief dip into sort of human research versus non-human animal research. I did kind of hint at the fact that things like gene therapy were a good segue into ethics, and this is where we come back to that. So we have this idea that gene therapy, the ability to go in and correct or adjust malfunctioning or non-functioning genes to produce the correct proteins, that could help us correct humans' disorders and diseases. We could try and fix things that are causing problems. But of course, there are always ethical concerns. And again, especially a lot of people are more concerned about things related to humans than other animals. So there's concerns about gene mutation or gene manipulation and how it might affect reproduction. We don't know if there are any long-term effects. Theoretically, it should only impact the one target site, but we don't know a lot about this new sort of technique, so it might be something that could have effects long-term. And then we also think about things like, okay, so if I manipulate someone's genes and then they go on to have children, their children's genes have that same manipulation because we changed that hard code in the parents, but the children have no way to say that they were okay with that. So they have a manipulated gene, a changed gene, an engineered gene, but they had no say in that. So that's really hard when informed consent is one of those big tenets of ethics in research. And then there's a lot of disagreement between different groups and sort of people in different fields as to how much gene editing should be permitted. Should it only be permitted if it were to correct, say, a life-threatening illness? I think we talked about Huntington's disease, where it's caused by one particular gene mutation. And if you have that mutated gene, you will at some point develop that disease. Well, what if we could fix that? We could save someone's life in the future by correcting that issue. Is that okay? Well, what about if we wanted to do something like correcting diabetes? Or what if we started talking about correcting things like autism? Where do we draw that line? Is everything worth fixing, or does it need fixing? And that is a massive argument that can be had. So if you have ethics classes, this is an interesting one to chat about. I took a philosophy of biology course, which talked about the ethics of what we can do in biology and what we should do in biology, and it was a very enlightening course. But for these things, there are no right answers, and it's still being heavily debated with lots of different people, lots of different groups having their own perspectives and their own agendas for this. So right now, gene therapy is a very gray subject, we can say. And also, while we're on the topic of maybe gray area topics, we can talk about stem cell therapy. And you know that this is another one of those hot button concepts where some people are really gung-ho to be doing stem cell research because it has the chance to correct what used to be uncorrectable issues. We can use stem cells to become or replace damaged cells anywhere in the body. So we can talk about pluripotent embryonic stem cells. So pluripotent just means that they can turn into anything. There are less free-form stem cells that you can get from adults. So adults have, especially I believe in our bone marrow, we have cells that are a little bit flexible, but they can't be absolutely anything. There are some restrictions. With embryonic stem cells, they can be turned into absolutely anything. And that's because most of the roles that our cells take on is determined by what they're surrounded by. What environment are they in, and what other cells are nearby. So if you took a stem cell and put it in the brain, that cell would figure out what signals it's getting from the surrounding area and be like, oh, we're brain tissue here. Great, let's specialize as that. Or maybe you put them in the spine and it's like, oh, okay, we have sensory neurons in this area. I'm going to be that now. So it's lots of promise for potentially fixing things like spinal disease and damage. But again, ethical concerns. So is it okay to be intentionally creating or using already existing embryonic stem cells? Is it okay to create embryos for research or not? And that's one that most people side on the not. And most countries have bans on using embryonic stem cells, but not all. So again, lots of debate in this area. And then there's lots of dangers, of course, posed by non-approved stem cell therapies that some clinics will offer. So this isn't something that is, they attempt to regulate it, but it isn't consistent everywhere. And if it's not heavily regulated, then people can do things that maybe aren't the best. So there's a lot of potential issues here as well. And again, we could spend many classes talking about these. But what we're going to do instead is come to our wrap-up for this chapter. And we can talk about the idea that from what we've gotten so far in this course, psychology has really advanced dramatically. And especially in the last couple of years, where we're concerned with the brain and how the brain works, we're getting a lot of advancement, a lot of technological advancements, a lot of advancement in understanding. And so we've gone from sort of a more simple start to something that's a lot more complex. And our methods have gone from sort of logical inferences and assumptions to being able to do things like actually sequence people's genes. But we still have quite a lot of methods that have carried forward. As we go on and as we get sort of this more sophisticated equipment, as we get sort of more complex methods, and we kind of hand-waved a lot of the complex math and modeling that goes on in the background, but even those theories are getting more and more complex. We're finding that psychology research gets more and more specific, so people will specialize on a particular area of the brain, and there's a lot less looking at the brain as a whole. So you might notice that trend in some of our upcoming chapters. And then with our discussion on ethics, we have lots and lots of knowledge, but we also have to keep in mind our responsibility as ethical researchers. So the responsibility that we have when working with humans and non-human animals. We have responsibility to people who might be helped or harmed by looking at some of these techniques. But we also have to accept that a lot of these things, there is no right answer. And that's why for at least now, we defer to ethical review boards to determine what we can and cannot conduct in research. And that's going to differ dramatically based on where you are, whether it's in between universities or even countries. They're all going to have different rules. So it's always good to know what you can and can't do before you get started. But I will leave it there for this chapter. You might have noticed the chapter quiz for this chapter is already up on Moodle, so you have access to that. And then next week we're going to start chapter 5, I believe. So that'll be our Tuesday. But I hope you all have a good rest of your week and a good weekend, because that's coming up.